So we're looking at the process of translation, and that's basically how do we go from that messenger RNA to a protein that was coded for by a gene. So the proteins that make up living cells are long chains of amino acids. They're held together by what are called peptide bonds. So we can refer to a long protein as a polypeptide chain. Now, remember that the base sequence in DNA determines the base sequence in RNA. So we've already transcribed the gene. Now it's up to the base sequence in in that messenger RNA to determine the amino acid sequence in the protein. So the codon is kind of the language of messenger RNA. It's a group of 3 sequential bases on messenger RNA that code for a specific amino acid. The anticodon is a group of 3 sequential bases on the transfer RNA that actually matches the codon on the messenger RNA. They would be considered complementary RNA pairs. So how does this work? First, the messenger RNA attaches to a ribosome. Remember, it's come from the nucleus that's already copied the gene in the DNA. The transfer RNA molecules then find their specific amino acids and bring their amino acids to the ribosome. The transfer RNA anticodon matches up to the messenger RNA codon using the complementary base pairing rules, putting the amino acids in order. The peptide bonds form between the amino acids, linking them in a chain because the transfer RNA gets released. And the transfer RNA just goes back out to the cytoplasm and gets another amino acid and comes back. It's just like getting more building materials. So once the stop codon is reached from the gene, that's when the protein is released. That's when the production is done. So when we take a look at the diagram here, we've got the messenger RNA molecule that's coming out from the nucleus, out to the cytoplasm, and all the way out to a ribosome. This ribosome is depicted using a larger blue piece, which is the larger subunit of the ribosome, and a smaller yellow subunit. Okay, there's 2 sections, and basically all we know about the ribosome is that it's designed to hold that messenger RNA and 2 transfer RNA molecules in place. So the transfer RNA molecules are these hairpin shaped, I'm not even sure how you describe those, type structures, and attached to each one is a specific amino acid. That's all they can bind to, okay? So they go out to the cytoplasm, they get their specific amino acid, and they come to the ribosome. Once the messenger RNA is in place with its codon, for example, this first codon is AUG. That's a start codon. The anticodon is UAC. See how they're complementary? That's what determines that methionine is the first amino acid to be put in place in that growing polypeptide chain. So before these 2 can shift over like a typewriter, almost like an assembly line, that peptide bond has to form here between methionine and phenylalanine. Then everything shifts over to the left. When that occurs, the next amino acid, the next transfer RNA molecule can bring in the 3rd amino acid, put them in place. Notice how it only shifts by 1, and that's so that way the complementary pairing can match up. So lysine is put into place, the next peptide bond has to form, and then everything can shift over, and a 4th transfer RNA molecule can be put into place. So you can see in the next screen, in slide D over here, that this is the growing polypeptide chain. This is all of the amino acids held together. And as the messenger RNA shifts over, the transfer RNA is released, so it can go back out to the cytoplasm and pick up a new amino acid and then come back. So let's take a look at how this works.
A protein is a chain of amino acids. The process of linking amino acids together to make a protein is called protein synthesis. Protein synthesis occurs in a cell's cytoplasm. mRNA leaves the nucleus through a nuclear pore after it is transcribed from DNA. The assembly of protein in the cytoplasm takes place on ribosomes. Ribosomes consist of 2 subunits, a small ribosomal subunit and a large ribosomal subunit. Floating within the cytoplasm are amino acids and transfer RNAs, or tRNAs. Each kind of amino acid binds to one specific type of tRNA. Notice the 3-base region on the tRNA. It is complementary to a 3-base region on the mRNA. Each group of 3 bases on the mRNA is called a codon. The complementary 3-base sequence on the tRNA is called the anticodon. A small ribosomal subunit binds to the mRNA at the start codon, AUG. A specific tRNA carrying the amino acid methionine binds to the mRNA. The anticodon on the tRNA matches the codon on the mRNA. The large ribosomal subunit now binds to the small ribosomal subunit and the mRNA. A second tRNA with its attached amino acid binds to the second codon of the mRNA. The first amino acid is transferred from its tRNA to the second amino acid, and a bond between the two amino acids is formed. The protein chain has begun to grow. The first tRNA detaches from the mRNA. The ribosome moves down the mRNA to the third codon. This codon is recognized by the anticodon of another tRNA carrying an amino acid. The growing protein chain is transferred to the third amino acid. The second tRNA detaches. The ribosome again moves along the mRNA. A codon is exposed. Again, the codon is matched by the anticodon of a tRNA carrying an amino acid. The protein chain is shifted, adding another amino acid to the chain. The tRNA that gave up its amino acid detaches, and the ribosome moves down the mRNA strand to the next codon. This process, called elongation, builds proteins one amino acid at a time. The 3 bases that make up a codon determine which tRNA attaches. Since each tRNA carries a specific amino acid, the sequence of amino acids in the new protein is determined by the sequence of bases in the mRNA. The ribosome continues to move along the mRNA. At the end of the mRNA, the ribosome encounters a stop codon, such as UGA. The completed protein is released. The tRNA floats off, and the mRNA detaches from the ribosome. The ribosome splits into small and large subunits.
So one thing we want to take a look at that we're going to practice is how exactly is the messenger RNA read. The messenger RNA, remember, is read in in codons. And so that's the series of 3 bases. So let's say we take a look at CCC. If that's the codon, it's an easy one to remember right now. Let's take a look at what that means. We look at the first base using the inside ring, okay, that inner circle. So I find the quadrant that's coded for by the C. So then I can read out the second base is a C, and the third base is a C. Therefore, the amino acid that CCC codes for is proline. Remember, this is the messenger RNA codon. If CCC is the messenger RNA codon, what is the anticodon for the transfer RNA? Hopefully, you said GGG. Now, do you see proline anywhere else on the messenger RNA wheel? Nope. So that means that there are only 4 codons that code for the production of proline. Now, once you've practiced working with the codon wheel, I want you to come up and see Mrs. Learned or your teacher and ask about how to use the messenger RNA table, which works the same way, but it's just a little bit different.